The next type of membrane-bound receptor system is called receptor tyrosine kinases. First of all, in the name there, tyrosine is an amino acid. It's one of a specific amino acid. And then a kinase is anything that adds phosphates onto something else. It's a kinase. If you remember, phosphofructokinase was one of the that was one of the enzymes, an important enzyme in glycolysis. But anyway, with this, the diagram they're showing here, it's another integral protein, goes through the entire membrane. It has a receptor on the outside, and then it basically has an enzyme on the inside, or an active enzyme site on the inside of the membrane. And these work, two of these together, and when they both get activated, or when they both receive a signal molecule, then they will end up bonding together. They call that dimerization. A dimer is typically when you have two of the same molecule that are being bound together. We'll hear that again later on with some genetic mutations that happen because of dimerization. But anyway, we get a dimer, and then that is that's the activated form of these tyrosine kinases. So we've activated these enzymes, and the thing that they're going to put phosphates on is each other, actually. So we've got these two molecules together. The one protein is going to phosphorylate the other one, and the other one's going to phosphorylate the first one. So we get to the end here where there's phosphates on all these different tyrosines. There happens to be 6 in this diagram, but all these different tyrosines on each of the proteins. So we've activated this whole system, we've got it phosphorylated, and we're going to get something called relay proteins, and a term that I have not really used yet, something called a transduction pathway. But basically, what we've done is we've gotten the signal from the outside of the cell to the inside of the cell, but it still has to be carried to wherever activity is going to happen. Typically, it's a series of things that happen that get the signal from the membrane here all the way into wherever the activity has to happen. That series of actions or series of steps that gets it there, that's called a transduction pathway. In this case, what we've done is we've got the signal to the inside of the cell, but now we have to launch this transduction pathway. And so we have these relay proteins that they've marked here. And essentially these relay proteins, they come along attached to the phosphorylated tyrosines, and that activates them so that they can pass the signal onto the transduction pathway. Okay, and so again, we've accomplished our main goal. We've got the signal from the outside of the membrane to the inside of the membrane, so that a transduction pathway can come and take the signal and continue to pass it down the line, which we'll learn about in the next video. So one last type of membrane-bound receptor system here. And this last one is called an ion channel receptors. This is the simplest one, I think. But basically, a signal is received by a gate protein that's on the membrane of the cell, and the signal basically just opens up the gate protein, opening up a pore so that some other molecule can fit through. The signal molecule is not opening this up so that it itself can get through. It's a different molecule here. But often what will happen, oftentimes it's an ion here, that's why it's called an ion channel receptor, but calcium or sodium are examples of that. And what they'll do is they will enter the cell, increase the concentration of themselves on the inside of the cell. A lot of times that'll trigger all kinds of different changes of proteins inside the cell. So if all of a sudden we have a whole lot more calciums inside of a cell than we normally would, then there will be proteins that will actually be activated or deactivated by that. So the signal, once received on the outside, we open this pore and it's going to change the environment on the inside, and that way our signal has actually entered the inside of the cell, because it's going to trigger some proteins to work, and some other proteins not to work. So those are all the different types of receptors that the book goes through. And again, and I'm going to say this a bunch of times, but again, these are just examples. These are some systems, there's other ones, and every single time there's a signal from one cell to another, every different case is going to be just at a different case. They're going to work differently. So anyway, the next video that we're going to get to, now that we've received these signals and somehow gotten them to the inside of the cell, the next step is getting them from they're inside the cell to let's get them to the right place in the cell so that things can happen. In some case, we have to take these signals and amplify them, they call it, or make them a larger amount of signal. Anyway, we'll get into that in the next video. Yeah, we'll see you then.